Mitochondrial DNA, mtDNA, is a unique type of DNA found within the mitochondria, which are specialized structures within cells responsible for producing energy. The formation of mitochondrial DNA is believed to have occurred through a process known as endosymbiosis. According to the endosymbiotic theory, mitochondria originated from free-living bacteria that were engulfed by ancestral eukaryotic cells. Over time, a mutually beneficial relationship developed between the host cell and the engulfed bacteria. The host cell provided protection and nutrients, while the bacteria supplied energy in the form of ATP, adenosine triphosphate, through a process called oxidative phosphorylation. During this evolutionary process, the engulfed bacteria gradually lost some of their genetic material, transferring it to the nucleus of the host cell. As a result, the host cell's nuclear DNA now contains most of the genes necessary for mitochondrial function, while the mitochondria retained their own independent genome, which is mtDNA. Mitochondrial DNA is circular in structure, similar to bacterial DNA, and it contains genes that code for essential mitochondrial proteins involved in energy production. However, over time, many genes were transferred to the nucleus, and now the majority of mitochondrial proteins are encoded by nuclear DNA. Mitochondrial DNA primarily encodes a small number of proteins: rRNA, ribosomal RNA, and tRNA, transfer RNA, molecules that are crucial for mitochondrial protein synthesis. Unlike nuclear DNA, mitochondrial DNA is inherited exclusively from the mother. This is because during fertilization, the sperm cell's mitochondria are typically excluded from the fertilized egg, and only the mitochondria inherited from the mother are passed on to the offspring. In summary, mitochondrial DNA was formed through a process of endosymbiosis, where ancestral eukaryotic cells engulfed bacteria that eventually became mitochondria. Over time, the genetic material of these engulfed bacteria was reduced, resulting in the formation of a separate, circular genome within the mitochondria, known as mitochondrial DNA.